Hello students, welcome back to a new video of Sunanda's tutorial. So in this video I will discuss the chapter 10 of 12th class NCERT biology book, which is Microbes in Human Welfare. So how microorganisms helps in different sectors. At first the microbes in household products. The common example is the production of curd from milk. Microorganisms such as lactobacillus and others, commonly called lactic acid bacteria, grow in milk and convert it into the curd. During the growth of lactic acid bacteria, it produces acids that coagulate and partially digest the milk protein. It also improves the nutritional quality by increasing the vitamin B12. In our stomach, the lactic acid bacteria play very beneficial role in checking disease causing microbes. It is a very important point. Then the dough which is used for making bread is fermented by using baker's yeast, which is Saccharomyces cerevisiae. Then toddy, which is a traditional drink of some parts of southern India, is made by fermenting sap from the palms. Then the microbes are also used to ferment fish, soybean and bamboo shoots to make foods. Then cheese, which is one of the oldest food item in which microbes were used. The large holes in Swiss cheese are due to the production of large amount of carbon dioxide by the bacterium named Propionibacterium cermani. Then Roquefort cheese is ripened by the growing of a specific fungus on them for a particular flavor. Then microbes in industrial products. The production of industrial scale requires growing microbe in large vessel called fermenter. Then fermented beverages. The yeast Saccharomyces cerevisiae used for making bread and commonly called as brewer yeast, because it is used for making the fermenting or is used for fermenting malted cereal, fruit juice to produce ethanol. Wine and beer are produced without distillation, whereas whiskey, brandy and rum are produced by distillation of fermented broth. Then antibiotics, these are chemical substances which are produced by some microbes and can kill or retard the growth of disease causing microbes. Penicillin is the first antibiotic to be discovered, and it was a chance discovery. Alexander Fleming, while working on streptococci bacteria, once observed a mold growing on one of his unwashed culture plate, and around which staphylococci could not grow. He found that it was due to some chemical produced by that mold, and he named it as penicillin after the mold Penicillium notatum. Later, Ernst Chen and Howard Florey made its full potential effective antibiotic. Then chemicals, enzyme and other bioactive molecules which are produced from microbes. Citric acid is produced from Aspergillus niger, acetic acid from Acidobacter aceti, Clostridium butyricum from produce the butyric acid, then lactic acid from Lactobacillus, then ethanol from Saccharomyces cerevisiae. Then enzymes like lipase, pectinase, protease, streptokinase also produced by different microbes. Then bioactive molecules like cyclosporin A produced from a fungus Trichoderma polysporum. Then stannine is extracted from Monascus purpureus, which is a yeast. Then microbes are also used for sewage treatment. This treatment can be carried out in two stages. One is primary treatment, one is secondary treatment. Primary treatment involves physical removal of particles from sewage through filtration and sedimentation. At first, the filtration can remove the floating debris or floating pollutants, and sedimentation, it removes the grit or soil and small pebbles. All solids settle, form primary sludge, and supernatant form the effluent. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment, the primary effluent is passed into large aeration tanks This allows vigorous growth of aerobic microbe into flocks. While growing, these microbes consume major part of organic matter in the effluent. This significantly reduces the BOD or biochemical oxygen demand. BOD is a measure of organic matter present in water. The greater the BOD of wastewater, more is its polluting potential. Once BOD of wastewater is reduced significantly, the effluent is then passed into settling tank and where the bacterial flocks were allowed to sediment. This sediment is called activated sludge. A small part of this sludge is pumped back to aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into large tank called anaerobic sludge digester. During this digestion, bacteria produce mixture of gases such as methane, hydrogen sulfide, carbon dioxide. These gases called the biogas. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. Then microbes in the production of biogas. Biogas is the mixture of gases produced by the microbial activity and which may be used as fuel. Then certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are collectively called the methanogens or methanobacterium. These bacteria are also present in rumen of cattle. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, these bacteria help to break down cellulose and play an important role in nutrition of cattle. Thus, the excreta or dung of the cattle, commonly called gobar, is rich in the bacteria. Is rich in this bacteria. Dung can be used for generation of biogas, commonly called as the gobar gas. Then the biogas plant, the technology of biogas production was developed in India mainly due to the effort of Indian Agricultural Research Institutes IARI and Khadi and Village Industries Commission KVIC. The biogas plant consists of concrete tank in which bio waste are collected and the slurry of dung is fed. A floating cover is placed over the slurry which keeps on rising. The gas is produced in tank due to the microbial activity. The biogas plant has an outlet which is connected to pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used as fertilizer. The biogas thus produced is used for cooking and lighting. Then microbes as biocontrol agent. Biological control of pest and disease such as ladybird to control aphid, dragonfly to control mosquito, then Bacillus thuringiensis to control wide range of insects, then Trichoderma fungi protect root system and control plant pathogen, baculovirus to attack insect and arthropods. Then microbes as biofertilizer. Biofertilizers, these are fertilizers that can be employed for crops or that can be applied in crops for better yield, and it can enrich the soil nutrient quality. The main source of biofertilizer are bacteria like rhizobium in the root nodule of leguminous plant. Some are free living like Azotobacter, Azospirillum. Similarly, the fungi which are mycorrhiza present in the root system of the plant of genus Glomus and absorb phosphorus and water from the soil for the plant growth. Then cyanobacteria which form a symbiotic relationship, that means Annavena with Azolla. Then free living nostoc, oscillatoria and blue green algae also. These are some cyanobacteria which are living in free condition in soil. So these are some biofertilizers or natural fertilizers which helps in increasing the soil quality or nutrient quality of the soil. So it is all about the microbes that can be used in human welfare. It is the chapter 10 of your NCERT biology book, 12th class NCERT biology book. I hope you really enjoyed this class. In my next class, I will discuss the chapter which is your biodiversity and conservation, and that is the last chapter because your exam is tomorrow. So I will upload that chapter by the evening. So please like, share my videos with other and subscribe to my channel Sunanda's Tutorial. Thank you.